One of the recent additions to Instant Designer has been the ability to have the gene cloned into a different vector other than the standard production vector, or PMX, with the choice of the antibiotic resistance. So in this example, I'd just like to show you this particular sequence here. We covered in a previous video that value and prime gene synthesis are available. This one isn't available for value, but I do have the option to fix this. In this particular case, if I hover over it, it says that repetitions are detected. So I can first start with optimizing the gene again. In this case, I'm interested in working with yeast. So I'm just going to put the shorthand in here for Saccharomyces. I can find that. And I know my auth range for this small area here, so I'm just going to put this in. So we have that there. So I click the checkbox. So here it's displaying the sequence and the protein sequence. The value is still not been fixed because we haven't optimized that. So in this particular case, hopefully this can be optimized, and yes, it can, as you can see. So if I jump back to the initial tab, I have the option for this particular one to have it as a value gene. So I can select that here. So as I mentioned, one of the recent additions is to change the vector within Instant Designer. So I'm interested, as I mentioned, in optimizing for the yeast system. So I believe that if we scroll all the way to the bottom, we will see this one here. So this is a vector which is perfect for that. So I can select this. And then the other thing that's been added here is the ability to do downstream plasmid prep. So you can see here that these options are shown with estimated yields. So I'm just going to be clicking the first one here. It's changed this now from research grade to transfection grade. So I click save. And as it takes care of this, you'll see the price changed. Again, these prices depend upon when the customer is logged in. So you'll see here now in this particular case, it's shown very conveniently the sequences here. It's shown that for this sequence, we have the option of having a value gene. It's going to take this long. And then here, this is the vector that it's in. And then this is exactly what we're doing. So it's a much simpler way in Instant Designer to be able to add this kind of add-on service. The customer also has the ability to flip back to, if they want to, a prime gene in this case, but in this case it's only allowing standard here. So I think here I'm going to be happy with just the value gene service. It's in the vector that I want.